Name of Allah. Today we are going to discuss the muscular tissue in detail. So skeletal muscle first. Let me tell you that the muscular tissue basically consists of cells and the associated or supportive connective tissue. Here in this picture you are looking into a bone and the tendon is being attached to it and continues forward. You see the transversely cut section of the muscle fiber. This is skeletal muscle here, and in which you can see that there is a whole bulk of a muscle being covered by a connective tissue which is made up of dense irregular connective tissue called epimysium. Then in the inner side of this epimysium, it is sending the septas inside, and what they are doing, they are basically enclosing or surrounding the bundles of the fibers or muscle fiber. You call them as vesicle. Here the name of this epimysium changes to perimysium, and then again you would be looking that the perimysium is again redividing inside, and the individual delicate the muscle fiber is enwrapped by a reticular fiber, a delicate layer of connective tissue now called as endomysium. So in this way you can see that the muscle, its bundles and its individual muscle fiber or cells, they are being covered by the three layers. The outermost is epimysium, then the perimysium and the endomysium. Let's discuss what they do. The function of the, you would say, the connective tissue which are holding this muscle. They have to perform two basic functions. Number one, they transmit the force mechanically which is being generated by the muscle contraction inside. This is required because the individual muscle cells actually do not extend from one end of the muscle to the other in a complete way. So that covering helps to transmit the force. Now, what next they do? They have to provide a passage for the neurovascular bundle. The rich capillary network, lymphatic vessels, nerves, blood vessels, they are supposed to enter this muscular syncytium so that they have to function in a very accurate way. Muscle could be classified as voluntary and involuntary. Under voluntary, you label the skeletal muscles, and under involuntary heading, you add the cardiac and the smooth muscles. And there is another way to classify or address them. You can label the striated muscles and the non-striated. The striated muscles include the skeletal muscle and the cardiac muscle, and then the other thing you add in the non-striated are the smooth muscles. Now we have to discuss a little bit about the embryological development of the skeletal muscle to know the reasoning of its multinucleation and the reason of its, you would say, the cell responsible for its regeneration. For this, you are looking into picture in which you can see a single muscle with the centrally located nucleus. What is going to happen? Actually, the skeletal muscles, they are developed or differentiated from the mesodermal or mesenchymal cells called myoblasts. What happens? They align themselves and fuse together to make longer and multinucleated tubes, now called as myotubes. Now they have to synthesize the protein to make the myofilament inside, and they appear as a cross striation under light microscope. So when there is a fusion of these myoblasts occurs, the centrally located nuclei then take the place in the peripheral region, and so many now nuclei appear as a multinucleated inside a cell which is very much elongated. And now another thing you can see here, that the few myoblast cells, they do not actually fuse but rather remain as a group of individual mesenchymal cells, and now they call as stellate cells, and you find them on the outer or external surface of your muscle fibers. So what they do, sometimes they are the reason to proliferate and produce new fibers of the muscle if there is a muscle injury, but the power of regeneration in the skeletal muscle is limited. Now nucleus on the peripheral region of the cell, and the myofilaments that appears as a cross striation, they take the very central position, because the major or important function of the skeletal muscle or the muscle is contractility. So the central location is allocated to the main myofilaments, and the nucleus takes the peripheral position to perform the functions. Now this is another view, very good view, showing the polygonal or hexagonal appearance of the muscle fibers or muscle fascicle. You can see inside that skeletal muscle. Here the connective tissue again covering you are looking into. Another important thing, that there is the highly specialized function of the cytoplasmic organelles of the muscle cells. So the terminology here is changed for them, like plasma lemma or plasma membrane, here called as sarcolemma, and you call it cytoplasm as sarcoplasm, and endoplasmic reticulum as a sarcoplasmic reticulum, and mitochondria here you can come across with the terminology of sarcomere. So keep in mind with all these changes which are important to discuss. Another good thing about the connective tissue and the muscle proportion is that muscle responsible for fine, highly controlled movement, like the muscles of your eye, extraocular muscles, what happens that they have small number of muscles or small fasciculi in comparison to the relatively greater proportion of the perimysial or connective tissue. Means less muscle but more connective tissue in your extraocular muscle. But in contrast to that, the gross movement involvement of the muscles, like that of the gluteal region, they have got larger bundles of fasciculi with little connective tissue, perimysial connective tissue would say. So much muscle and less connective tissue, and this whole perform a very good function in relation to the size and the particular muscular movement concerned. Now we are looking into the again cut section, and here we are gonna discuss about the various cross striations, that how they appear. You are looking into a picture showing a sarcomere, in which you are looking the Z line on both sides. What actually they are, looking into a picture showing the sarcomere, and as you know that the sarcomere is basically the contractile, in the contractile apparatus, it's a functional subunit, and where it is present, you are looking into a picture of cross striation, in which you see the alternating light and the dark bands. The dark bands are A bands, you can see in the picture, and the lighter bands are the I bands. The A bands represent the anisotropic or birefringent in polarized light microscopy. And what about the I bands? They are isotropic, means do not alter the polarized light. That is why they are showing different shading, the dark band and the light band. And from one Z disc to the other Z disc. This is the extent of your sarcomere. In this, you are now looking into a thick filament in the center, and from the each Z disc, you can see that the thin filaments are running parallel to that of the thick filament, and again the A band you have seen in the very middle. There is a zone called H zone, in which there is no overlapping of the thick and thin filament, and it's in very middle being dissected by a line that is called as M line. And what else? In that very the region of the thin filament, you can see that there is the on its edges the Z disc. So in you can say sarcomere, two Z lines should be there to complete it as a whole sarcomere. You can see the myofibrils that are present inside the muscle cell of fiber, which are specialized contractile element holding inside the myofilaments, which are contractile protein, and another name for that thick and thin filament are the myosin and actin filament. If I can able you to show the picture in an enlarged view, that you can see the thick filament or myosin having two identical heavy chains, you can say, and they are twisted together as myosin tails, and their rod like appearance are running parallel and twisted in like a form of a braid, and or like a rope, and on its one end they are having a head on each side of the heavy chain. And this is basically you can see in the lower picture as well, that the actin filament will also be showing the type of protein, the tropomyosin, then you can see the troponin, and there is another accessory larger protein, namely titin. These are also present along with this all. You can see two pictures. The above picture is showing the muscle in its relaxed state, and the picture that is below, you can look into a contracted skeletal muscle picture. In the very relaxed state, what is going to happen, that the thick and thin filaments, they are not completely overlapping each other. So whenever there is a region of the eye band which is showing lighter stain, that tells us very clearly that it is the portion where the thick filament do not overlap the thin filament, which is why eye band stain more lightly, you would say. And we can compare it with the contracted muscle phase. Here the thick and thin filament, they completely overlap each other. From here we are very much clear that the length of this individual thick and thin filament does not vary or does not change. It is just the sliding of these two that makes the muscles to contract individually. A little bit about the contraction cycle. Here in the very first picture, you are looking that the repeated contraction cycle involves 5 steps, you would say. In this phase, you are looking, the head of the myosin is going to be attached to that of the actin molecule of the thin filament here. Stage 2 of the second stage of contraction cycle, you can see that there is a release of the myosin head, which is being uncoupled from the thin filament. Now you are supposed to look into the third stage. In the third stage, what happens, that the bending of the myosin head occurs. As a result, ATP hydrolysis advances a short distance to the thin filament, And now towards the fourth stage, which is you call as force generation phase, in which myosin head releases the inorganic phosphate and then the power stroke occurs. Then look into the fifth stage, in which reattachment in the last stage occurs, the myosin head that tightly bends to the new actin filament here. Now in this very picture you are looking into the synaptic knob, and here what happens, a nerve impulse triggers the release of acetylcholine from the synaptic knob into that of the synaptic cleft. The ACTH or acetylcholine binds to the ACTH receptors in the motor end plate of the neuromuscular junction. And what happens now, they initiate a muscle impulse in the sarcolemma of the muscle fiber. Now, as the muscle impulse spreads quickly from the sarcolemma along the T-tubules, and the calcium ions are now released from the terminal cistern into the sarcoplasm. And now what are these T-tubules? They are basically, let me tell you that the sarcoplasmic reticulum is arranged in a series of repeating network around the myofibrils, and this system, which is called as T system, having numerous tubular invagination of the plasma membrane. Each one is called a T-tubule. It basically penetrates to all level of the muscle fibers and are located between adjacent terminal cistern or at the AI junctions. What I want to say is that in each sarcomere, two T-tubule and the two terminal cisterns are present, and they are forming the triad in the skeletal muscle. And here you can see the release of the impulse along with the release of calcium ions, and they use this T-tubule system as a basically road for the passage of their signals, so that the contraction takes place evenly among the whole muscle fiber. Now look again in this picture that what is triad, what is T-tubule, what are terminal cisterns of the sarcoplasmic reticulum, and how longitudinally you can very well see the Z line and A and I band, and all the structure is very much clear around the muscle cells. You can visualize them. Now a little bit about the innervation of the muscle fibers. Here what is going to happen, that the myelinated motor nerve fibers, they branch out within the perimysium, and they are supposed to reach the muscular fiber, and the axon from a single motor neuron can form this motor end plate on one or many muscle fibers they reach. And innervation of a single muscle fiber by a single motor neuron, they will provide the precise control of the muscle activity, like in the extraocular muscles, which is very much important for the eye movement. And the larger muscles with coarser or rough or gross movement have motor axons that typically branch profusely. Almost 100 or more muscle fibers are being innervated by them. In a sense that a single axon and a muscle fibers in contact with its branches make a motor unit. And you can also see that the number of motor units and their sizes control the intensity and precision of the muscle contraction. Now you are looking into a picture of the sensory receptors, I would say, of the striated muscles. Here you can see the intrafusal muscle fibers and the extrafusal muscle fibers. They act as a proprioceptors, and they provide the central nervous system with all the information collected from the musculoskeletal system, and they perform the function of stretch detectors, and you call them as muscle spindle, being enwrapped by the perimysium. And what else they have got? They are flattened cells layer filled with the interstitial fluid and a thin muscle fibers with nuclei in it called intrafusal fibers, and several sensory nerve axons you can see penetrate each muscle spindle and wrap around individual intrafusal fiber. And if there is any change in the length or distension of the surrounding muscle, which you call as extrafusal fibers, that is caused by the body movement and being detected by the muscle spindles, and information is taken to your spinal cord. So different types of sensory intrafusal fibers mediate reflexes to maintain postural and other activity of the opposing muscles, which are very much mandatory in your walking mechanism. Now you can see in the picture that the cross-striation are very much visible in the longitudinal section of the skeletal muscle, and below you can see the transverse section in which peripheral nuclei also present there around the muscle cell. And if you want to look into a chart form of the skeletal muscle, you can write the parameters on the left like a muscle cell. You can mention the length that of the cell in along with its diameter as well, and then the location. You can mention all the muscles present. They are also form the part of your organs like tongue as well, the diaphragm and the esophagus, and rather the extraocular muscle in addition to your muscles of the locomotion. And connective tissue are looking into, we have already mentioned the 3 types, and fibers are single because they are basically non-branching, and striation is very much present prominently. Nucleus are multinucleated, T-tubule system is definitely there, we have just mentioned, and definitely non-branching, so no cell-to-cell junction you can find. Specialized features are well-developed smooth endoplasmic reticulum and the T-tubules, and being voluntary, so it's a type of their innervation, being voluntary, so somatic, and type of contraction is that they have got all or none lower here, and regulation is through the binding of calcium and causing the tropomyosin movement, and exposes various sites on the actin filament. We have already mentioned the stages, and growth and regeneration they have mentioned. In a sense, mitosis is none, but a little regeneration through the stellate cell we initially mentioned, and if there is any damage, so predominantly hypertrophy occurs. So that's all about